All right. Hi, folks. Welcome to today's program schedule. My name is Angela DiCiccio, and I am president of the Surfaces and Biomaterials Foundation this year. I'm a hardware engineer at Verily Life Sciences and really excited to be with you this afternoon. On behalf of all the members of the planning committee, our communities at the Surfaces and Biomaterials Foundation, and the American Preclinical Society, our collaborators on this project, I want to extend a warm welcome and thank you for dedicating some time from your day towards participating in our first live virtual conference. We're delighted you're joining us and encourage feedback and interaction throughout today's program via the chat box, the poll, and the attendee profile features of the Crowdcast platform. Explore, please let us know what you think. Your input is highly important to our ongoing success, supporting collaboration and thriving through virtual platforms as we transition and seek to empower new modes of communication, collaboration, and innovation as in-person options aren't available at this time. Normally, we would be having this meeting in person as well as 2020 biointerface workshop and symposium. However, this year we'll be moving online. So check out our website, surfaces.org, if you're inspired by today's program to learn more about our upcoming virtual symposium, which will take place throughout the month of October in small segments in different weeks. Today, we're excited to focus on critical technologies that undoubtedly will influence everyone here. That's sterilization technologies and its relationship to COVID, but also materials and devices. This year is truly a unique time to focus on how surfaces of biomaterials impact our world and set the foundation for our future. I think we're all becoming a lot more mindful. Our program today will feature speakers from Nelson Laboratories and Medtronic, who will discuss how novel materials and alternatives to ethylene oxide sterilization have developed and been transitioned throughout the massive learnings and efforts initiated and transformed by our global COVID response. Much like our ability to advance these fields, coming together to enable productive, proactive, and inspired communication is not possible without key collaborations and partners. So as we open up our program, I'd like to take a moment to introduce Mark Smith from American Preclinical Society, our collaborators here.
Here we go.
Awesome.
Now a word from your sponsor. Thanks, Angie. Good afternoon. My name is Mark Smith. I'm the former Chief Scientific Officer from American Preclinical Services, now associated primarily with the business development team. I wanted to welcome everybody, as Angie already did, to the open house. We appreciate the opportunity to be associated with the Surfaces and Biomaterials Foundation. APS has been working with the Foundation since 2009, and we've hosted several of these sessions over the years at our campus located in Coon Rapids, Minnesota. Unfortunately, this is as best we can do right now, but we're all excited about this process, and we'll see how it works. Just to get a quick overview of our facility and services, APS is a full-service preclinical research and testing facility. We're located in suburban Minneapolis. We have major focus supporting manufacturing and development in the medical device industry. Company's about 230 employees, and we've been fortunate to stay close to full operation during all the recent events. I think it's been a very fantastic opportunity to see how the continuity of things within the healthcare and specifically this component of the healthcare business has been going. APS works primarily in large animal models for surgical and interventional studies, as well as offering a full range of ISO 10993 services for biocompatibility testing for medical devices. In addition, we have a new analytical chemistry team specializing in extractables and leachables testing. And we will be hosting a virtual website here coming up on June 25th at 11 a.m. You can go to the APS website to check into that and for registration. But we just wanted to kind of give a plug for one of the new processes. We're going to learn a little bit from how this one works, and we will continue on with that as a continuation of some of these virtual get-togethers. With that, I'll turn it back over to the speakers, and thank you very much for participating.
All right, we saw the paper.
Keep going.
Great. Hi, Ingrid. So I need to share my screen. Yeah, so welcome everyone. I'm the past president, Bill Tylacker, and I'm here today to help stimulate the economy. We're gonna have a trivia session. We're gonna give away 4 25-hour Amazon gift cards. And so everyone's eligible to participate. There's a polling feature at the bottom of your Crowdcast screen. There'll be a series of 7 questions. If you fill out those questions with the correct answer, you'll be selected into a random drawing, and we'll announce the winner at the end of the webinar. So question 1, what is the website of Surfaces and Biomaterials Foundation? SIBF.com, surfaces.org, surfaces.com, biointerface.org, or biomaterials.org. How many years has Surfaces and Biomaterials existed? That's going to be a tough one. I know some of you will get it and some won't. Where is the location of Biointerface Symposium 2021? What is the name of the Surfaces newsletter? Another challenging question. Who is sponsoring the open house? Who chaired the inaugural Surfaces and Biomaterials Symposium in 1991? And I'm not sure who knows Larry Ferrari, but people on the East Coast might know who that is. And question 7, what is not a benefit of becoming a member of Surfaces and Biomaterials Foundation? So that's the trivia. You'll see 7 questions at the bottom. Please fill those out, and we'll announce the winners at the end. Thank you.
Thank you, Bill. Next up, we would like to jump into today's program. I'd like to introduce Nick Symes from Nelson Labs. Fun fact, Nick will be celebrating his 6-year anniversary on Sunday, and he met his wife at an 80s night at a dueling piano bar. Adventures certainly to be remembered. Nick?
Yes, that's true. All right, thanks, Angie. I'm just gonna share my screen here. All right, I hope everyone can see that. So good afternoon, everyone. Thank you to Bill and Angie and Ingrid and Joshua and everybody at SIBF for this opportunity today. When I was first approached about Nelson Labs being involved in this open house, I really jumped at the chance. Just with the increased focus right now on PPE, our protective barriers group at our lab has just seen an enormous increase in requests for testing. And the creativity and the ingenuity that we've seen from folks trying to combat this pandemic has been amazing. So we're happy to be a part of this discussion today. What I'd like to do right now is just briefly tell you about Nelson Labs, who we are, what we do, and how we can help. So a little bit about myself. Again, my name is Nick Symes, Regional Sales Manager. I live in Minneapolis. I've been with Nelson since August of last year. I have roughly 10 years in the testing industry, from food to pharma and medical device. Have my bachelor's of chemistry from the University of Minnesota. And another fun fact, I was just accepted today to University of Iowa's MBA program. So pretty excited about that. There's my contact information. That's my cell. So if you have any questions once this is over, please feel free to contact me, text, phone, email, whatever's easiest. So what is Nelson Labs? What do we do? I really like what our VP of Ops, John Bolander, says whenever he's asked this question. So I'm going to steal his response. He says, whenever you're in a hospital, anything that is in you, on you, or around you, Nelson Labs has probably tested that for safety and efficacy. So whether it be through testing or consultation, Nelson Labs is really here to safeguard global health. A little background about Nelson Labs' history. We started in 1985 by Dr. Jerry Nelson and his wife, Linda, based in Salt Lake City, Utah. We've grown significantly since then, now with 2,500 plus active clients in 57 different countries, with performing over 700 different microbiological and analytical tests. We service clients in the medical device industry, pharmaceutical industry, and the tissue industry. So again, focusing on safety, efficacy, safeguarding global health. A little bit here about our parent organization, Sotera Health, which was established after Sterigenics' acquisition of Nelson Labs. We're 3 companies working together, Sterigenics on the sterilization side, Nelson Labs on the testing side, and then Nordion, which is that 3rd company. They're based out of Canada. They procure cobalt-60 for use in sterilization. So the 3 companies all working together with one common goal. Here's a look at our global facilities. Like I mentioned, we're based out of Salt Lake City. We have standalone labs in Itasca, Illinois, Nelson Labs Europe, which is in Leuven, Belgium, and a new acquisition of ours, Gibraltar Labs, which is in Fairfield, New Jersey, one of our pharmaceutical centers of excellence. And then we also have what we call integrated labs, which are labs that are embedded in a Sterigenics sterilization facility, and those are located all throughout the world. A quick look here at our various accreditations and certifications to be able to do what we do. ISO accredited, FDA registered, drug and device, as well as DEA certified for controlled substances. One thing that really sets Nelson Labs apart is our participation in industry committees. As you see here, a total of 27 delegates on AMI, ASTM, and ISO working groups. So when you work with Nelson Labs, you have the confidence to know that you're working with industry leaders and that we're not only just following these standards, but we're active in the decision-making process and helping to drive these standards forward, ensuring the safety of end users. So I don't have enough time today to go into depth into each different lab area, so I'm just going to do my best to highlight a few key areas here. We're all here today because of our interest in sterilization. So looking at Nelson Labs core competencies here, you can see one of the things we do best is to support sterilization validations from a variety of different modalities, as well as supporting ongoing QC lot release tests to ensure sterility assurance. On the product validation side with reusable devices and cleanability studies and healthcare processing, and of course, protective barrier testing, so important right now with masks and gowns and drapes. And also a variety of supporting services like biocompatibility, in vitro performed at our facility, and we support in vivo studies through external lab partners, as well as extractable leachables, a very hot topic right now. And we also perform packaging testing. And then another thing we have is our expert advisory services group. Our consultants are able to provide regulatory support through study design justifications, acceptance criteria, toxicological assessments, and even facility validations. So what I like to drive home today is just the fact that Nelson Labs can offer you a truly end-to-end solution. So no matter where you are in your product life cycle, we're here to support that testing and provide you the data and the insights you need to make decisions, from product design through regulatory submission to QC lot release testing. So that's CliffsNotes version of Nelson Labs. If you have any questions, there's my contact information again. So please feel free to contact me anytime, phone, email, text, whatever's easiest. Thank you again for this opportunity, and thank you to SIBF. So I'll turn it back.
Cool. Thank you, Nick. And sounds like you will have a very delightful weekend of a lot of things to celebrate. We hugely appreciate your participation both in the Surfaces group as well as Nelson's support of our programming today, and your expertise within the field. The activism of Nelson and your group is huge. And with that, I'd like to introduce Martell Winters, who will be speaking to us next. Martell is a talented specialist in microbiology as well as radiation. And he's been with Nelson Labs for 24 years, starting in the radiation sterilization group. He was responsible for bioburden testing and radiation validation studies. Martell's superpowers include microbiological process validation, radiation sterilization of medical devices, allograft tissue, and pharmaceutical products. He's also a member of many of the AMI sterilization standards working groups that Nick was referencing, and a U.S. representative to ISO for 3 working groups. Outside of his inspiring work on global committees and as director of microbiology at work, Martell is an enthusiastic advisor to his wife's microbiology experiments in the kitchen at home. Recent developments and achievements include yogurt, kombucha, sourdough, and kimchi. With that, please help me welcome Martell, and let's learn.
Thank you, Angie. Okay, let's get my screen going here. Okay. Is everything showing well?
Yes, we can see your screen.
Okay, great. Thank you. Normally, I always enjoy presenting. Normally, presenting is easy for me because it's usually on topics that I've worked at for decades. I've been at Nelson for 26 years now, and this was a little bit different for me because this whole concept of decontamination and sterile processing of single-use PPE is new to all of us, starting about in March or so, as far as an industry-wide type of thing that to have to consider. So it's been a steep learning curve for me and for others as we've tried to understand how to take single-use PPE and adapt our typical or even non-typical sterilization methods to these products. So I want to start with a little bit of history. So we started getting a lot of inquiries at the beginning of the year, and then it really hit hard about in March regarding validation of mask reuse. And Nick mentioned we have a protective barriers group, and that group about in March just got hit with tons of mask testing. So at this point, we're running 24/7 for mask testing just to try and keep up with demand. So there was really no established process for how to go about this. FDA fairly quickly came up with some emergency use approval documents, and I'm going to go through a couple of those. And then a lot of discussions ensued. It felt to me like almost my entire month of March was focused on these discussions with all these different groups and trying to develop appropriate test methods and determine which sterilization methods or decontamination methods are likely going to be best. So in a month's time, we had all these discussions with that list there: J&J, FDA, the Nursing Association, WHO, and all those other ones, and some others I'm sure I forgot. And intellectually, it was stimulating. It was really interesting, but it was quite a challenge. So internally, we created this COVID-19 Sotera task force to start to put the information together. So maybe just big picture first. So typically, the big concern initially was masks. We're also seeing an issue now with other types of PPE, particularly gowns, where we're seeing some shortages there. But the first hit was really on masks, and so that's the bulk of my experience on this topic. So we kind of have the three masks. So we have the surgical mask, and the primary focus on a surgical mask is to protect against large droplets, splashes or sprays. And when you perform the filtration efficacy testing, the minimum requirement usually is about 30%. The respirator, these are the N95 masks that we've heard so much about for the last few months. And those are validated to a much better efficacy at 95%. Now we have this emergence of all these cloth masks now that are going around everywhere. And how can those be reused? What are their efficacies? And the efficacy, of course, varies largely. However, we've done a lot of testing on some of these homemade masks by larger corporations that are starting to make them. And we see many of them fall within about between 70 and 80%. So they actually work quite well. Better than a surgical mask, but not as well as a respirator. Now, to hit the EUA document, so the emergency use approval document. So the first one that we focus on a lot within Nelson and as we worked with customers was the one regarding face masks and respirators. There were two main areas of focus here. One was to make more single-use masks available. And then the second was to allow for reuse of single-use masks where it's appropriate. So I'm going to focus on the reuse of single-use masks component here. FDA said that they'd help to expedite evaluation processes if you could provide this information. So I'm going to go through what information they're requesting. So first, they want a description of the process. What are the cycle parameters? And they're requiring a CI, a chemical indicator, as well as a BI, a biological indicator. So the next is a validation of the bioburden reduction or disinfection. So the requirement they put in their document was they wanted equal to or greater than a 3-log reduction of virus, specifically some form of coronavirus, and greater than or equal to 6-log reduction of mycobacteria or spores. So mycobacteria is a bug that causes tuberculosis. It is very resistant because of its normal cell structure. And spores, of course, we know how those are very resistant as well. They also talked about soils on the mask, either that the soils be removed or that you validate that they don't interfere with the reduction process. So, of course, chain of custody and safeguards, as you're collecting masks at the healthcare facility, as you're shipping it over to the reprocessing facility, and as it's coming back, you want to make sure that you're not contaminating the masks or that the contaminated masks aren't contaminating people. And then material compatibility, make sure that materials don't degrade with multiple reuses or re-sterilizations or decontaminations, and to determine a maximum number of reprocessing cycles. Of course, filtration performance, that's critical with repeated use as well as a fit test. And the fit tests are not such a big deal with surgical masks, but with the N95s, that's where we see that being really important. So we got together with some friends at Johnson & Johnson, and we approached FDA and said, we have a handful of questions as we are guiding customers and providing guidance to the industry. We want to understand what flexibility we have on some of these things that are brought up. So, for example, we want to know about sample size. So there's a European document, 14683, that uses a sample size of 5 for each test. And we felt that was appropriate, and we checked with a couple of FDA representatives on the phone, and they agreed that seemed reasonable. We also wanted to group the masks. As we're trying to figure out a flow of testing masks, we wanted to have these big picture groups. And we felt that it was appropriate to group them into either do they have a valve or not a valve, and then different general mask type, mainly cloth versus some type of like a spun polyethylene. I'm probably getting the polymer wrong on those. And so they felt it was appropriate for us, as we're setting up these validation approaches, to group them like that. Now the BI and CI part. So a lot of UV and chemical exposures, many of them are not in a chamber. We're accustomed to having biological indicators and chemical indicators when we have a chamber in place to help understand the environment within the chamber. But in some of these decontamination methods that were being looked at, like UV and maybe spraying with a chemical or something like that, that's not in chamber. And the representatives on the phone were not that excited about having a parametric release kind of approach, meaning they did not want to see something where we just said, well, we expose it to this amount of light at this distance for this much time. They really want to see some type of biological and chemical indicator that it's functioning correctly. We talked about that could we use published literature to avoid having to gather actual data on viral inactivation data. The viral activation data can be difficult to obtain sometimes, and I'll show on the next slide here something to discuss this more. But we were wondering, okay, if there are substantial data in the literature to support that what we do have data on is more difficult to eliminate than viruses, would that be okay? And they felt that could be reasonable depending on what you provide. So this is a general scale for germicidal processes. It's very well understood. It's been, they actually, FDA reproduced this in their other EUA document regarding sterilizers and disinfectant devices. But it's been in books and generally understood amongst microbiologists for I don't know how many years, 20, 30, well, more than I've been involved, so 30 or 40 years. So this idea being that you have at the bottom, meaning the least resistant, you have these lipid or medium-sized viruses. And then as you go up the pyramid, they get more robust with regards to resistance to germicidal processes. And you see the lipid or medium-sized viruses at the very bottom. So coronaviruses are a lipid virus. They have an envelope around the virus, and it makes them not only very susceptible to germicidal processes, but even susceptible to just drying as it sits on a surface. If you've looked at the CDC information, you'll see that all the data that we have to date suggests that if not in a matter of hours, at the very longest on some surfaces by about 3 days, that the viruses are they're no longer active. And so as you work your way up the scale here, you'll see the ones that we're talking about in the FDA document are bacterial spores and mycobacteria. They're at the very top of this pyramid. Now, you would expect perhaps some kind of commingling of resistance if they're right next to each other. For example, between vegetative bacteria and fungi, they're right next to each other on the pyramid. You might expect there to be some overlap depending on the species, which might be more resistant than others. But when you have the spores and mycobacteria on the very top of resistance, and then the coronaviruses at the very bottom, clearly they are easier to kill. And that is not only is it generally understood, but FDA in our discussion with them felt that was appropriate. If you had solid data on germicidal processes for spores or mycobacterium, that you would be okay to not gather empirical data for coronavirus. And then they actually have, this is a quote from that same document I referenced in the previous slide that says that very thing. So, because sterilization processes render devices free from vital microorganisms, including spores, and because disinfection kills most recognized pathogenic microorganisms, it can generally be inferred that sterilization and disinfection should minimize the viability of the coronavirus. Okay, so a couple more questions we had for them regarding this disinfection or sterilization of masks was, okay, generally speaking, to be able to kill mycobacteria or spores, they're usually fairly harsh chemicals. They are not the type of chemicals where you would want to immerse a mask in it and then fairly shortly after that be wearing it. There are a lot of residuals on those masks. They're smelly and they're fairly caustic. They're not terribly biocompatible. And so we said, okay, what about, would there be some conditions under which you might be happy, you might accept an idea of a low-level disinfection? So if we're going back here to this pyramid again, vegetative bacteria are one step above lipid or above the coronaviruses as far as resistance goes. And so even if we just show low-level disinfection of this typical panel of microorganisms, Pseudomonas, E. coli, Klebsiella staph, and Enterobacter, would that be sufficient? Mycobacteria can be tested, but again, it uses these harsh chemicals, and also it often takes a lot of incubation time. So if we're quickly trying to get validation data, mycobacteria can impede the ability to get data quickly. And FDA said that they felt that was reasonable. It was reasonable to focus more on a low-level disinfection and not have to use harsh chemicals if the mask is for the same user. And we felt that was a reasonable answer. Mycobacteria, like I said, that's the tuberculosis bug. If someone had tuberculosis, it was wearing the mask, and then if someone else is wearing that mask, conceivably, you could potentially, I suppose, be exposed to that. So if the same user, if you have if the mask is being used by the same person, there was some flexibility there to not have such stringent requirements for reprocessing of masks. The last one we had was regarding cleaning of the masks. Cleaning blood or other body fluids or makeup, which seem to be the biggest issue on a lot of these masks. To be able to clean those from masks is very difficult, and that really reduces the efficacy of the mask. So we proposed, instead of having to show cleaning, what if we just say that there's a visual inspection performed, and if there's soil on the mask, then those are not reused, and they felt that was a reasonable approach. So just quickly, how do we do this challenge? So how do we measure decontamination? As far as the microbial load goes, what you see here on this picture, there's some material, the mask material there with droplets of microorganism. And, I mentioned already the requirements of the mycobacterium and spore reduction. And will novel methods, decontamination methods, be able to meet those criteria? And that's a really valid question. That was one of the big questions that we were trying to answer, was what methods can we find that are amenable to the material in the mask, but still will give us appropriate log reductions for microorganisms? So questions like, do you have to do some type of simulated use to simulate the physical damage that occurs just by wearing a mask? Can we just use new materials, or do they have to be washed, for example, or used? So these are the types of questions we're trying to figure out. Big picture, when we look at decontamination methods, the ones, these are both fairly well known, both the sterilization list and the decontamination list. So hydrogen peroxide, EO, radiation, moist heat. What we've seen as far as a good portion of the EUAs that have been granted by FDA are utilizing vaporized hydrogen peroxide. It seems to be an effective method. And for many mask materials, it seems to be fairly easy on them. For decontamination, so that's the sterilization column. For decontamination, dry heat, microwave, UV, ozone, liquid like IPA, those are all fairly well understood and used on surfaces and on some medical products. But it was new to have to deal with these on masks. Now, FDA has been really responsive, and FDA gets a bad rap sometimes because of what their role has to be. And but I have a number of friends at FDA that I interact with on a regular basis through writing standards, and they are people, and they're actually nice people. So they just have a huge undertaking, and they're always balancing this risk. So they've got this. They're always trying to balance, okay, what is the risk of allowing these other things to happen versus the benefit of having more PPE available? And in a fairly short period of time, just to list those, you can see all those different EUAs that they have granted for different topics. So they've been really working hard to try and be responsive to the industry's needs. So just a little bit of data I thought I'd provide on that we've gathered over in the last little bit on reprocessing masks. So we've done some testing with IPA sprays. The IPA has been very effective, of course, at killing microorganisms, vegetative microorganisms. And we found that it reduced the efficacy of an N95 to about 85 to 90%. So instead of 95, they went down 5 or 10%. Now, that was a single use there, a single reprocess. But that was fairly well soaked. That mask material was soaked after those sprays. The issue, and this is something I did not know because masks has not been my specialty in the past, is that the N95 rating, that they get an extra bump up to N95 usually based on this electrostatic charge that the mask material has. And as you reprocess those, mainly when it hits liquids, then that will reduce or eliminate that electrostatic charge. And then you only have the material itself that is causing the filtration efficacy. So that does eliminate quite a few sterilization methods that use very high humidity or actual moisture, because it does make it harder to still obtain the good data on the masks afterwards. We did some work with radiation. We have a large number of irradiators around the world. So we tried 25 kilogrades, a standard radiation process at cold temperatures. And we found that at a single dose of 25 kilogray at cold temperature reduced the efficacy from 95% to about 90%. We do not have exhaustive data yet, like how many times you could do that. But 25 kilogray is really more than you would need to be able to meet the criteria that FDA has established. We just shot at that dose just to get an initial feeling for how things looked. It's been interesting to and I need to do a quick time check here. So I should be done. Angie, should be done.
You have 10 more minutes, and then we'll take 5 minutes for questions.
Okay, well, we're right on track then. Thank you. So, so the World Health Organization reached out to a number of companies, and we've been fortunate enough to be involved with this discussion for several months now. And so their goal was they wanted to gather data on validation of processes that would easily be usable in third world countries. So they figure the people here in the U.S. that and Europe will focus on the big picture decontamination with big chambers and stuff like that. But what about all these small hospitals or doctors' offices around the world that don't have access to all this equipment? So they were looking at some fairly simple things. So they were originally looking at 4, and that was cut down to 3. So and 2 of those are the ones that are specifically for these third world countries. So UV light was originally being looked at. That ended up being cut out because it was too much to fit in with everything else that they were trying to gather. Although initial data support that it's a fairly effective method, with the one issue of penetration not being super good. So methylene blue plus light. This was new to me. I'd used methylene blue in science experiments as a. Sorry about that. I forgot to disable my IM system. I'd used methylene blue before, but I did not realize that methylene blue in the presence of light actually has a fairly well understood bactericidal component or activity. So it's really simple. You just spray the methylene blue on the mask material and expose it to light, even sunlight, and then it has a fairly decent log reduction. Now their focus was only on viruses. That was their primary concern in gathering this initial set of data for WHO. Then also dry heat. Now dry heat is often thought, again, a big chamber, very high temperatures, but in this case, we're looking more like the 80 to 90 degrees Celsius, which actually is very effective against viruses. And then vapor hydrogen peroxide, which if you've been keeping up, and on the sterilization stuff of masks, that's been the more common method for reprocessing of masks. So testing is underway. On the first slide, you saw, in addition to me, I had Alpa Patel. She's one of my associates at the lab. She's our point person for all this testing that's being done that we're involved with. So it's really interesting to see how WHO has jumped in here trying to gather data, and I've really enjoyed the approach they've taken in finding simple ways to decontaminate viruses on masks that hopefully anybody in the world can use. So when we're talking about face masks, our main focus here with 3 main things. So is it still, so what is the efficacy of the decontamination method? Is it still functional, of course, and in the safety of the user? And within Nelson, we kind of put together this general flow chart of how we envision the validation going of these methods. So I'm not going to go through every step here, but big picture. So first, how does the material function? Does the material function correctly after the decontamination processes? Before, of course, so we can have something to refer to. And then we have a disinfection and sterilization component down at the bottom there. And then after multiple disinfection and sterilization uses, or implementations? Does it still meet the requirements? And then lastly, is it biocompatible after this either disinfection or this sterilization process? And I'm not going to go into a lot of detail here, but just so you're aware, these are the general criteria that people are looking at in the industry. So ASTM has a number of test methods that they refer to, as does the European nations. So the European document there, a lot of similarity there, but there are some differences. But these are the types of tests that are being applied and the general criteria that are expected. Now, the N95 masks are the ones you hear of most often. They're level 1. And for levels 2 and 3, the requirements there are actually at 98%. So there are these other masks that are sometimes used, but the N95 tends to be the more common. And as you know, it's been the one that's most been talked about. I'm going to talk briefly just on the biocompatibility side. So normally biocompatibility is a fairly lengthy and expensive process to make sure that you're addressing all the biocompatibility needs of both the product, the product itself, as well as the patient. So but what FDA has been trying to do here is say, well, from a risk-based approach, what makes sense? And what we don't want to have happen is after these masks are decontaminated, that they're getting bruises or blisters or rashes on their faces from wearing these. So what we've seen working well with FDA regarding biocompatibility is perform a cytotoxicity test. That's a fairly typical first test, the first approach to use. But then to review the mask materials and what the parts of the mask that are touching the face, what are those materials? Are there any known sensitizers or irritants on there? And then a fairly simple risk assessment with those materials. And with the one exception that if the mask is going to be on the market after the pandemic as well, then they're going to expect a more robust approach. But for right now, because of the low risk there, they've simplified the biocompatibility requirements with regards to reuse of masks. You probably saw just a couple of days ago, there are certain respirators that are no longer authorized to be decontaminated for further use. I'm not going to go through this in a lot of detail, but they're due to issues they were seeing with the masks. There's a list of those that no longer, they're still approved for single use, but they're not approved for reuse. And this is just one of those things that as FDA gathers data, they're trying to provide those data to the industry so that we can make better decisions. There's been a lot of recent publications on sterilization and reuse, disinfection of masks. And the hard thing is that it's always difficult to interpret these publications. The questions like, which tests did they perform? What were the critical variables in those tests? And it's very difficult to get the full story, the full story from publications. So just as a scientist, I always say, be careful and going too far with interpretation of data that you read from, especially from a single article or something from somebody. So that's it. I welcome any questions or discussion that we'd like to have.
We do have several questions, and I can go ahead and ask. One person, Daniel Worthington from Verily, asked in the chat, could you give an example of CI and BI indicators used?
Sure. So what we're used to seeing as far as CIs and BIs, for example, in a steam sterilization cycle or an EO sterilization cycle, the CI, there are different ratings there of CIs. One is just a gross indicator of whether it saw heat or whether it saw EO. So it's just a color change of the tape from one color to another. The more complex CIs involve things like wraps in them, and still sometimes a color change, but inside a challenge, so that you're actually showing that the sterilant is penetrating through a package or a wrap or something like that. like a Bowie-Dick test as an example of that in steam sterilization. For BIs, a biological indicator, usually, again, those are just on a simple filter paper, and it's inoculated with usually more than 1 million spores of a specific microorganism. And then after the sterilization process, then those BIs are placed in growth medium, and we just look for growth. Did something grow, or were there surviving microorganisms after that process? And that's what makes it hard in looking at these more unique methods of sterilization. Is oftentimes there is no established chemical indicator, and there's no established worst case microorganism to use as a biological indicator. Or if there is, it still is difficult to place on a substrate and in the decontamination area to actually represent the product itself. So there's some additional challenges there.
Okay, we have several more questions. So it's acceptable to just decontaminate masks if used by the same person to diminish waste mainly.
I'm sorry, can you say that again?
Yeah, so it's acceptable to just decontaminate masks if used by the same person to diminish waste mainly.
Yeah, so that's the information we're getting. Is if it's the same user, there is information we're seeing is that they're amenable to the option of not applying a full sterilization process between uses. The risk of since the viruses are the easiest to kill, and since it's the same person wearing it, they're not going to reinfect themselves. So the main concern is what's happening with the outside exposures to the mask. And since the viruses are so easy to kill, then that has been, we understand, has been an acceptable approach. Although the default does tend to be sterilization, as you see in most of the EUAs, they're focused on a full sterilization cycle.
Okay, and then another question: do you think who would consider UV light if compelling data came out? The World Health Organization.
So yes, they would. They actually broke up into groups, and were having groups that were drawing information from the literature to see what was available. And they really did want to try UV light because, again, they felt that was a fairly simple thing that any hospital could get. And it really came down to a financial issue with just procuring masks and being able to pay for the testing and things like that. It just became too difficult, so they ended up having to drop that one. So yes, I think they would enjoy be happy to get more information on that they could provide to hospitals.
Right. Another question: why is coronavirus a 3-log reduction when it's easier versus a 6-log reduction for the more difficult organisms?
That is a great question. What I think it is, and there's still not a lot of data on that. At least it's been a few weeks since I checked. I think the reason is probably, I've not asked that specific question, but scientifically I can see a rationale for that because of 2 things. One, because it does die fairly quickly on its own. When it's not in its host cell, it is not, like most viruses, it is not very stable. So the requirement could be less. The second is that the current information that the most recent information I saw is that in a droplet of sputum, for example, since viruses again are associated with host cells, they tend to not see a super high count of viruses in that small area of say a droplet. So I think that their thought was the level of contamination is likely less, and the fact that it dies so quickly also makes it to have the requirement be less. That's my best educated guess.
Right, and I apologize if some of these you've already answered. I'm just kind of reading the questions, but how high the temperature can be used for dry heat decontamination without damaging the mask material?
So a lot of these mask materials are some type of a polypropylene. And polypropylene does very well even in steam sterilization at 132. So it actually, there's some data that I've seen that support even up to 100, 110, it still functions quite well. And I've not seen that pushed to failure, so I'm not sure at what point you see failure there, but it can handle fairly high temperatures. But the reason that the data that WHO is exploring, the reason that's low at lower temperatures is because there's no need to go higher for the viral decontamination they're looking for. So the temperatures closer between 80 and 100 are very effective against viruses.
Okay, we have time for one more question. Would you be willing to write up a summary of the approach Nelson took to organize testing approaches, communication with the FDA, and developing a reprocessing flow to feature in the next Surfax, which is the Surfaces and Biomaterials newsletter? I'll unmute Angie, because that was a wonderful question.
Angie, that's your question.
Maybe.
Yes.
We can work on it.
Let's talk. We do have some of these.
Yeah.
Some aspects of what I talked about are already available on the website or on material that we are sending to customers. So it would be fairly safe. Let's talk after, and I think it'd be fairly simple to pull that information together in a cohesive way that then we could make easy for distribution.
Fantastic. Yes, that was inspired by your talk and everything that you've just shared with us. And hopefully the community here agrees. And if you do, go ahead and type in the chat box or give us feedback. But I think exactly what you said, this information is out there. And I think that's something a lot of us have found in this entire experiment. I mean, I've mentioned fun facts about people. And I think my fun side fact is I've been trying to become an expert in microbiology in my evening time. When you described that month of March and the amount going on, you're correct. It was so mentally stimulating. And I think I slept on average 3 hours a night because I was just thinking all the time about disinfection, sterilization. I'm a polymers kid. And so this is literally at the forefront of like, what's happening to my polypropylene? So thank you, Martell. That was an awesome overview of a lot of approaches. Very interesting. And we have a lot of questions. And if people have follow-up questions as well, please enter them into the chat box or the questions and we'll get them addressed later. Next, I'd like to take a moment to invite Mallika Kamarajugadda, a senior principal scientist in the corporate science and technology group at Medtronic, with expertise in materials research and selection in the areas of metals, surface modifications, thin films, characterization, and material structure property relations. So huge breadth. She has 20 years of engineering research and development experience with a track record of innovation and sharing of knowledge through publications in peer-reviewed literature. And in her current role, she leads programs in materials that are critical to enable next-generation medical devices and technology. She also partners with all of Medtronic's businesses as an internal technical consultant and researcher with subject matter expertise. So I'm very excited that Mallika will be here today telling us about ethylene oxide sterilization alternatives for medical device applications. Mallika, I'll hand it over to you to fill our brains with knowledge. Thank you.
Thank you for the nice introduction, Angie. Let me just share my screen here. Can you see the presentation?
Not yet.
Okay, once.
Oh, I see you're presenting your Crowdcast screen.
Okay, so can we see now?
Yes, I'll go ahead and focus that. It's on the actual PowerPoint, so if you want to go ahead and start the slides. There you go.
All right, good. So good afternoon, everyone. My name is Mallika Kamarajugadda, and as Angie introduced, I am a material scientist at Medtronic Corporate Research Group. I would like to thank the organizers for giving me this opportunity, and Just a disclaimer first, after hearing from the real sterilization experts, that I am a novice in the field. I'm not a sterilization expert or a microbiologist. So what I'm going to do today is give an overview of the different technologies from the materials lens and talk about some of the challenges and opportunities we have in this field, especially in the current environment and challenges. So this is just an overview of my presentation. I would like to take a few minutes on walking back into the history and then trying to look at the requirements from an FDA perspective. Then we can just go through the technologies that were developed, compare it with the EO sterilization, and then talk about some of the material science opportunities and then challenges for the selection and compatibility. So what this slide shows here is a brief history of the sterilization, starting back from the Egyptian time frame when natural available material was used to embalm bodies like pitch, tar, and aromatics. And then back to 300 BCE, Hippocrates, who is also known as father of healing, he acknowledged the importance of keeping the wounds clean, and he used substances such as boiling water to disinfect them. However, the first attempts of using heat for disinfecting or sterilization, the credit can be given to a Roman practitioner, Galen, and he was known to boil the instruments prior to taking care of the gladiators. The interesting thing, however, is that none of these people implementing these practices had any idea of why heat would reduce the infection rate. So let's just fast forward to the second half of the 17th century, when a significant discovery was made, which paved the way for sterilization as we know it today. And that was the existence of microorganisms and the discovery of that. And then we go back and then we can credit the discovery of first autoclave to Charles Chamberlain, who it is the first pressure steam sterilizer or autoclave. And then slowly through the time in the 19th century, we then go and we develop different modalities of sterilization. Low temperature techniques came. However, one thing that is of significance is to point out the contributions of Dr. Spalding here. In 1968, he developed an approach, systematic approach for sterilization of medical devices. And how do you classify the materials? He divided them into three categories: critical, semi-critical, and non-critical. And his classification is still used today. Now, if you are looking at this slide, for my own purpose, I wanted to first understand, and we have heard before too in the earlier presentation, decontamination or disinfection and sterilization. So from a perspective of understanding what the difference is, first thing to notice is they both do not ensure the same security level, and they don't necessarily inactivate all forms of microorganisms. The most important thing here being the bacterial spores. So disinfection destroys most of the pathogens and makes them inert. And typically it's accomplished using a chemical agent. Whereas sterilization completely eliminates all microbial viability and kills non-pathogenic and pathogenic viruses. And just for fun, another term that is becoming a norm these days is sanitization. And that I think is defined as a process of making usually an inanimate object clean. So again, the choice of disinfection and sterilization is done according to the risk of how the infection is spread. So again, we go back to Spalding's classification, and then we say that for critical medical devices, which are intended to come into contact with sterile tissues, some of the examples would be surgical implants, syringes, needles. Sterilization is mandatory and it is highly required. And then we go to the second category, the semi-critical devices. And some of the examples of those would be endoscopes and some that come into contact with mucous tissues, for example, the ENT instruments. And for them, the recommendation is trying to use a high-level disinfection or sterilization. And then we have the low risk, non-critical classification of devices or instruments, and examples would be the blood pressure cuff that you can use, and then these only low level of disinfection is needed. So how do I now quantify the sterilization and disinfection? So one way to look at it is trying to see how much microbes were killed. So there are two ways that the microbiologists use to quantify these. One is known as the log reduction, and this measures the percentage of microbes that are eliminated. And this graph here shows the percentage of colony of the reduction of the pathogens, and this is typically quantified in terms of a log scale. So when I say I have a log 1 sterilization, disinfection, I mean that I'm reducing the colony to 100,000 bacteria with a 90% reduction. So as we can see, a 3 log kill for sanitation would give a 99.9% kill. And for all the sterilization requirements, which we quantify as 6 log kill, it's 99.999% efficiency. Another way to look at it is trying to just plot it here as trying to see how many of them are surviving due to the sterilant action, since we know the death of rat is exponential and it can never be zero. So I try to quantify qualitatively here, plot the surviving microbes as a function of exposure to the sterilizing agent. And then here, you can see that the rate of death is never zero. And then if you take the log of these survivors versus time, that gives us a very important plot, which is used as a standard for sterility assurance, and that is SAL. It is the probability of any viable microorganism that is present on a unit after sterilization. So this figure basically tells you that there is one single item in a batch that is non-sterile after the entire process. So if I say I have a SAL of 10 to the power of -3, it means that there is a 1 in 1,000 chance that an organism is surviving the process. And if I say I have a probability of 10 to the power of -6, then I mean that it's the lowest probability, and this is used as an industry standard and assessment, as we know that the goal is to have the lowest probability. Then FDA tries to classify the sterility basically from the ISO standards as the state of being free from viable microorganisms. And you look at it holistically, the sterilization process is a complex process, and it starts and ends with the patient. It impacts the material selection, the processes that are used to make your device, the manufacturing, and also the distribution of these devices across the entire spectrum. So here you can try to classify the 8 important characteristics that are highlighted throughout the sterilization process. And then the top 2 refer to the biocidal activity that we have already talked about, and seeing that any process or the sterilization sterilant that is used should be able to quickly achieve the required limits. And then the next 2 categories that we have here are the strong penetrability, material compatibility, and these are interrelated, and then these kind of dictate how we choose our device. Material compatibility, as we know, is only it should produce a negligible change in the appearance or the function of the processed items, and then the packaging materials, even after repeated recycling. And when we say non-toxic, it means that there is no toxic health risk either to the operator or the patient, and it doesn't pose any hazard to the environment. And then the last 3, basically adaptability, cost, and monitoring, can be tied up to the last chain of manufacturability, where we want the process to be monitored easily and accurately with having both physical, chemical, and biological monitors. And then it should be cost effective too, along with being adaptable. Now, having looked at all this, now if we try to look at how we can categorize the known sterilization modalities, one way to look at it is trying to look at it on the base of the nature of the sterilant and how it reacts with the microorganisms. So you can classify them as physical or chemical processes. Physical processes, as we can see here, are basically either the ionization, radiation, heat, and then chemical processes are all those either be in the gaseous or the liquid form. And sterilization processes basically act on microorganisms either in chemical or physical way. For example, if we take steam sterilization, it is like more or less a physical pounding. The condensation of steam on the surfaces releases a lot of latent heat, and this energy damages both the metabolic and the structural components of the molecule and then makes it denatured. That's the same mechanism even that happens with the radiation modality too, both whether it is a ionizing or a non-ionizing portion of the radiation. When we go to the chemical processes, I would like to look at it based upon whether the chemistry is an oxidative based chemistry or an alkylating agent based chemistry. This categorization actually helps us understand more about the material's compatibility when we are looking or comparing different technologies. As you can see, the ethylene oxide, which is like one of the most dominant mode of sterilizations that was used, comes as an alkylating agent. And what that means is the high energy hydroxyethyl, it replaces the atoms in the membrane and then basically creates holes by adding carbon chains and then renders the whole microbe ineffective. On the other hand, oxidation is when you are looking at the basic atom and then it loses an electron. And since atom is the building block of the molecule, it chemically is ripping apart. And more, the other thing to take into account is also these oxidative agents further can also be energized by using additional radiation. And what that does is creating these high energy radicals, which are much more reactive, and they can actually do the killing by attacking the membranes. Let's take a brief look at the technology evolution of how these sterilization processes have developed in the spectrum here. And most of these processes that have been traditionally used for medical products include steam, ethylene oxide, or gamma and E-beam radiation. However, we can see that over the past 3 decades, there has been a push or more incentives to be able to develop the new technologies, and there have been several emerging processes. And one of the reasons there could be the decline in the use of ETO in the hospital, and also there is a result of increased concern over the toxicity of residuals for workers and how these residuals also affect the patient. Another reason I think which is increase in the use of minimally invasive therapies, and these minimally invasive therapies are made up of very delicate combination of different materials, and they required use of new and low temperature sterilization technologies. Along with that, somewhat the materials portfolio has also improved and increased, and these I think are some of the drivers for developing these other low temperature technologies. Lastly, again, trying to look at how we categorize the methods here for medical device in a manufacturing setting based on FDA guidance. FDA tries to categorize the methods as established or novel methods. And again, in established, you have category A and category B. What goes in category A are those methods that have a long history of safe and effective use, and they are demonstrated through multiple sources, and all of them have like recognized methods and standards for development, validation, and routine methods. And whereas in category B, you have these methods. FDA has recognized some, there are no dedicated standards for but then there is enough published information on the development and validation, and somewhat a routine control is available. So basically these would come as 510K approval for sterilizers, or a parametric based approval is what it is, the language is, where you have a certain set of process conditions and sterilization load, and the process is approved for that. And then some of the novel methods that are described here in the third column, like the vaporized paracetic acid, high intensity light or POTS, and even using just the sound waves, these are all in the infancy, and then they are all in the technology feasibility or R&D phase. Now here, this slide is actually trying to capture what the current state is. If we look at GUDID, the acronym stands for Global Universal Device Identification Database. We have more than 2 million medical devices listed in that. And out of those, 40% of them are provided as sterile to users and patients. So as we can see from the chart here, 50% of those are sterilized with ETO. And then these range from general healthcare instruments such as dressings all the way to our pacemakers, heart valves, dialysis sets, and vascular stents. And then gamma radiation is another commonly used industrial mode for medical devices. And it's actually considered favorable for use in processes because of its short processing time and better penetration. Also, compatibility with a wider variety of materials, although it is less than ETO. One of the bigger advantages of using a radiation-based method is that the process parameters that you see here, gas concentration, temperature, humidity, or exposure time, we don't have any of those process parameters that would affect your material compatibility. And then the radiation dose is pretty well established. And these sterilization doses that have been approved, they don't have enough energy to cause any radioactivity in the material that is known. Steam, although has been developed back in 1930s, it is still used for less than 5%. And that's because of the limitations on using steam for materials, polymers, which are heat sensitive, and it is used on some of the metals, but then corrosion is one of the problems for using it on the broad spectrum of metals. Although one of the advantage that came out of trying to use this is development of like heat resistant polymers to some extent, and also trying to identify some of these surface modification technologies to prevent the corrosion on the metals. So now we come and try to characterize, basically, this is just a table showing what are some of the important parameters that one should consider among the established methods if you want to choose your mode of sterilization. And here is where materials compatibility is coming into picture. The other things are more for validation, process development. But then when you are looking at the material compatibility, you can see that most materials are unaffected by temperatures. And that's why ETO has become such a dominant mode of sterilization for our industry. And as I pointed out earlier, based upon these 4 methods that have been well established and recognized by the regulatory bodies, we have for the past like 40 years tried to build our portfolio and then choose only those materials that are compatible to these modes of sterilization. Then just wanted to spend a few minutes on comparing the pros and cons of ethylene oxide. And the commonality here, as you can see, is ethylene oxide being like a small gas molecule. It has excellent penetration properties, and then that's what we have taken advantage of using in our devices, and especially the long lumens. And then it just kills about everything. And that's also a disadvantage. It just kills about everything. And it's pretty gentle and compatible with most materials and packaging. And some of these factors also have paved way into why ETO has been a dominant mode for over a period of time. One of the cons, however, is a really, really long time it takes due to the aeration requirements here, sometimes like more than a day. And again, this is trying to compare the variables. As we can see, ETO is become a highly controlled process. And then the advantages, as you can see, is you can have, it is used in a variety of settings. So you can have almost compartments up to 30 plus pallets to hospital-based settings. The adaptability is also one of the reasons why this is like widespread in the healthcare industry. One of the things then I would like to point out here is a typical sterilization process profile that describes a mechanism of how EO is working. And the first step in the preconditioning phase, where you are requiring the load to be brought up to a specific temperature and humidity level. And these are very essential for the proper sterilization. The important thing here to note is the humidity requirements. 30 to 80% of humidity range is preferred for optimum sterilization effectiveness. And this actually can also be, the requirement also can be traced back to one of the things that was pointed out by the earlier speaker on the spores, which are the most resistant bacteria to be killed, and somehow maintaining this humidity level tries to just swell up the pores and then just break them. And that's why maintaining a humidity level of 30 to 80% is optimum for the sterilization effectiveness. And then after the preconditioning and the sterilization process, it requires basically you remove the oxygen to allow the EO gas to fill the chamber. And then to be able to do that, there are a number of safety measures and controls, and there is a continuous monitoring of parameters, temperature, pressure, gas concentration, and time are done throughout the process. The third step and the most important step is the aeration phase. And this is why this is where the product has completed the EO process, but still it has some absorbed residuals. And during this phase, additional processing is performed to reduce the residual levels at a minimum to the required standards. And this probably is a highly controlled and monitored phase, because if there is any shift in the process, you might see an increase in the EO emissions. And then this slide actually just shows a snapshot on the chamber sizes that are used for different stages of the EO processing, the preconditioning, the actual processing chambers, and the aeration. And as you can see, we can actually load more than 30 pallets of fully assembled and packaged devices here. Then one of the issues that this process also brings is maintaining the logistics. From moving from one stage to the other stage, the load is being constantly moved. And then, so this brings in like logistics of transportation and trying to maintain the particular conditions that are needed to reduce the emissions or just prevent like the toxic emissions in the atmosphere. So that is one of the risks that is involved with the EO processing. And this actually is a good segue to look into how the sterilization landscape has been evolved. And ever since like ETO was moved as a list of known carcinogens in 1990, there has been an increased scrutiny in the usage as a sterilant. There have been alternate methods have emerged, and then these methods have gained momentum recently. However, as you can see that there has been a movement of trying to just social movement triggered by the cancer clusters that was formed, which most of you probably are familiar with the first plant closure in 2019, Willowbrook in Illinois. And as of today, I think there are at least 7 closures that have been in the news because of the increased emissions of the EO or identification of those zones as cancer cluster. And this actually has created an increased pressure even on the FDA and the regulatory bodies to come up with incentives to shift the market from EO as a primary mode of sterilization. And then the reason why I tried to bring this slide here again is, let's try to look at it. And then this was our status quo on how we developed our whole portfolio of materials, and then try to choose the materials that are compatible with the known sterilization. And then now with the incentives and the increased pressure to look into more alternative technologies, the compatibility becomes very important. And then this also gives us an opportunity to look at some of the materials that have not been used because of the restrictions posed by these 4 sterilization methods. As a starter, if you try to look at the materials compatibility chart here, this is where this graph is produced from the AAMI materials data, which is an excellent resource for looking at the compatibility rating for all the materials in the category. And what I have tried to do is try to look at the known methods, and then the compatibility rating is plotted out across the group of materials that we are using in our medical devices: thermoplastics, thermosets, adhesives, elastomers, metals, and ceramics. And as we can see here, and we discussed here previously, EO has excellent compatibility, good to moderate. However, if we try to bring in some of these low temperature alternatives like hydrogen peroxide, ozone, or hydrogen peroxide based ozone, that's when you try to see the compatibility rating is changing from medium to fair to excellent. Now let's try to we can look at the same slide in a different way.
We are at time, so if you could just go ahead and finish up.
Okay. What I can do here is trying to look at the materials here, basically break it down into different modes of materials that we have here that we use for making our medical devices. And highlighted are some of these: nylon, polyurethane, polysulfides, and cellulosic material. If you look at the first 3 columns, which are like the standard mode, the compatibility is rated as moderate to very good. However, when you are looking at the new technologies that we are trying to adopt, sometimes there is no data, and sometimes it is ranging from poor to excellent. So what this gives us is opens up the opportunity to be able to look at these and understand exactly the material stability as a function of these different sterilization modalities. Here, metals typically we say are mostly compatible and don't have any issues. However, one of the things we have to keep in mind is trying to look at these vapor-phased hydrogen peroxide or nitrogen dioxide or ozone. Most of these are oxidizing agents. And we know that some of them can corrode the metals. And it's like a delicate balance of trying to achieve the sterility assurance level without having to degrade your material performance. And that's where I think this changing landscape and the opportunity we have here to look at the new technologies opens up a window of looking at the compatibility basically from the fundamental materials perspective. Here is what we have, just trying to look at some of the literature that is published on the medical grade plastics, looking at the tensile strength after various doses of exposure. And this is known, and this is what we have been using for making our portfolio. However, this is an interesting and encouraging data here on the right side when you are looking at the tensile strength of some of the plastics after the vapor phase sterilization. You could see that I can go up to 25 cycles on some of these without actually a decrease in the tensile strength. That is an encouraging news, but then it is only on a certain set of material. And one of the opportunities we have as material scientists is to be able to expand this database and then try to understand the mechanical properties, the chemical stability, and also the surface degradation of these materials using these new modes of sterilization. And this is actually just one of these compatibility slides that a colleague of mine sent, which I thought would be interesting to this audience on the surface modification. Most of the medical devices we use have some kind of surface modification for adding a functionality or improving the performance. So this research here looks at a very thin polymeric coatings on silicon substrate and then trying to subject it to different sterilization technologies and assessing the hydrophilicity, looking at if there is an adhesion issue or if in general the coating has deteriorated and if the protein resistance is also deteriorated. And as you can see here, peroxide probably doesn't work well with some materials, but however, the coating here is only a few nanometers thick. So if we are looking at trying to establish a database on devices and surface modifications here, this could be a starting point, and then we can expand and build the portfolio of trying to look at various modifications that we use and use it to develop and expand our portfolio. And this is my last slide here. One of the things that I wanted to just highlight is device compatibility is what we generally think about when we are looking at evaluating a new sterilization mode. However, in this slide, we can see some examples of how complex our material build is, looking at even the metals with different surface modifications, metals in combination with polymers, as you can see in the in stents, as well as a lead that is used has a combination of electrodes, metal wires, as well as polymers. So instead of trying to put the whole thing and trying to look if the device is surviving the sterilization, we can actually break it up into individual materials and then try to look at the reaction kinetics. Again, as I pointed out, some of the new technologies work on oxidation. So, and then the other important factor is we are shifting away from ETO. So here the sterilization is accompanied, done by vapor phase instead of a gas phase. So condensation and the absorption and how the diffusion is happening, those are all the questions that we have to look at and then try to evaluate it at a fundamental materials mode. And also, we know that we already talked about peroxide sometimes is used for accelerating aging. Now, when you are trying to think about using the peroxide for sterilization, does it have a long-term stability effect and how is it going to degrade? So some of the questions that we have also provide like an opportunity for us as material scientists to be looking at this at a fundamental level. And I would like to close with just this. FDA has taken this innovation challenge here. And as you can see, this is also identifying the methods and technologies where it has already selected some of these technologies and trying to look at the compatibility of our portfolio. And whereas the other one is trying to look at the existing portfolio and how do you maintain the market share. So both of these offer opportunities in innovation and development, both from a chemical engineering, so that's materials chemistry, materials physics, as well as materials in general. So I would like to just close with this one here that summarizes basically the new mechanisms of action with the processes and how the materials interact.
Great. Angie, I have you on unmute. Do we have time for some questions? Did you want to read them or would you like me to?
So in spirit of the timing, I want to give a giant thank you to Mallika and your team for sharing all of this, like your approach and everything was amazing. We do not have time for questions. I know that we had booked until 1:30 with folks. And so, or rather 3:30 for the people not on the West Coast. So Mallika, may I ask, if we send you the questions, would you kindly write responses to them and we can feature that in our next Surfax and make sure that people get them with their slides?
Absolutely.
Thank you.
I'd be happy to do that.
That would be so awesome. Your knowledge is like just so expansive. It's hard to even to fit it in because I could just listen to you for hours. So thank you so much. I want to thank everyone for joining today. This was a really, really exciting chance to host our first virtual conference. As we wrap up, we really welcome any feedback on the platform, on your experience today, things that you would like to see. If you'd like to get involved in our upcoming events, we have a Surfax issue that will be coming out, and we're calling for articles or advertisements to go within there, nuggets of knowledge. In addition, we'll be doing our virtual Biointerface 2020 online, and then in 2021, we'll return to Portland. Before we go, I just wanted to thank our sponsors, Nelson Labs and the American Preclinical Services, our co-hosts, for making this event happen today. And for those of you who fought tooth and nail on the trivia questions, thank you, Bill, for putting them together. We have 4 winners who will get emails at the end. So, yeah, and I'm going to try my best on your names: Yelena Gorlin, Youngjin Q, Sebastian Rihanna, and Benjamin Tang. Y'all have check your email boxes for both the recording of today as well as your your trivia prizes. And with that, thank you. Please send feedback and have a wonderful day.